All right, we're covering Chapter 7, Section 4. This is take 2 because it screwed up and didn't record it. We're covering facilitated diffusion, diffusion, active transport, hypotonic, hypertonic, isotonic, and endocytosis and exocytosis. So pay attention. I'm going to be kind of quick because I want to, it's the second or third time I've had to do this because it keeps screwing up. Diffusion is the net movement of particles from a high concentration to low concentration. It's just simply put, looking at these diagrams, one side's red, one side's blue, but by physics, they move. They just move, okay? Over a long period of time, they're going to move so much that they're perfectly mixed together. That's diffusion. There's no real energy that's required because the particles are already in motion. Diffusion, like that, eventually you reach this equilibrium. And in that dynamic equilibrium, again, it's not necessarily, its concentration is now equal both ways. It's more along the lines of, okay, these things are moving, but they're moving at such a rate that they stay at an equilibrium. That's why it's dynamic equilibrium. In diffusion, we have the same things happening with the plasma membrane. Water can diffuse across the plasma membrane, but most other substances can't. When water diffuses across, that's just simple diffusion, versus facilitated diffusion uses transport proteins to move ions and small molecules across the plasma membrane. Can't remember my last thing I was saying, so I'll just start over. Water diffuses across the membrane freely. Facilitated diffusion is where we need the objects aren't big enough or small enough to go through the membrane. So what they're going to do is go through a protein. Most of the time, that protein is facilitated diffusion doesn't require energy. It just kind of connects and just flows back and forth. It's specific to everything, though. There's no energy required. Versus there's the diffusion stuff, but we'll watch it in class. And then we have osmosis. More specifically, with osmosis is the diffusion of water. So osmosis is where water diffuses across a selectively permeable membrane. In that diffusion, only water's moving. So what happens is if you have a bunch of molecules on one side, and you have a bunch of water in there as well. Let's say on one side, okay, let's say over here we have a ton of water, but over here we have a ton of molecules. Water is going to flow across to equalize the number of molecules in water and molecules of water. We'll try to have a dialysis lab where you see dialysis working like that. Probably use some eggshells to show it. You'll see it's pretty cool stuff to do. So we'll figure it out next week. Water is a solvent in a cell in its environment. It moves across the membrane until the concentration of solute is the same on both sides. Realize it's concentration, not actual amount. It's concentration. Remember that. The same thing with water. It moves until the water's concentration is equal on both sides. So if there's a ton of solute on one side, that means there's less concentration of water. And there's more concentration of water on the other side and less concentration of solute. So it's going to move until that's equal. So we have three different things. The first thing is isotonic. Isotonic solutions are where water moves. It's a dynamic equilibrium. It moves in the cell and out of the cell at the same rate. So it's a really happy place for a cell to be. Your blood is an isotonic solution. Hypertonic solution is where you have a lower concentration of solutes on the than the cytoplasm of the cell. So since I have a lower concentration of solute outside the cell, that means the concentration of water is more outside the cell. And since that's the way, that means my concentration of water inside the cell is less, and the concentration of solutes inside the cell is more. So what's going to happen is water is going to move into the cell to bring it to equilibrium because only water can move across. And in that movement, that water pressure inside the cell is going to build because it's like a balloon. It's like you keep blowing it up until it's full and it's going to pop. Animal cells do that quite often. Not quite often, but in a hypotonic solution, they do it very often. They blow up. Plants love it. They call it turgor pressure. Turgor pressure is when a plant has in a hypotonic solution, causing the leaves to be nice and full and out there. Versus, I believe the next one's hypertonic. Hypertonic solution is where you have more solute outside the cell than inside, or more of a higher concentration of solute outside the cell than inside the cell. That means you have a higher concentration of water inside the cell versus outside the cell. That hypertonic solution right there is going to cause water to flow out of the cell versus in. When that water flows out, eventually that water itself is going to shrivel up. Plants don't handle it that well. It's called a wilting process. It's where you see plants, you put them out outside on a hot summer day in the morning, you don't water them before the day. And that hot summer day, by the end of the day, they're actually going to start wilting. They're going to start losing the rigidity of that leaf. Then we have our osmosis in various solutions. Try to watch that to help better understand it if you have issues. Active transport. Active transport is where you actually do have to use energy. It's where you want to move something from a high concentration to a low concentration, such as like kind of moving a ball. If you push the ball down a hill, there's no energy required. It's going to roll down the hill. If you push a ball up the hill, it needs energy. Same concept. Think of it that way. Low is at the bottom of the hill, low concentration down here, high concentrations up here. We're trying to push it up to high. Why do we want to push it to high, though? Push it to high, we'll get to it in a second. An example is a sodium-potassium ATP-ACE pump. ATP-ACE stands for an enzyme that breaks down ATP. It's connected to the pump to help break off this phosphate. ATP stands for adenosine triphosphate. Tri means 3, phosphate means phosphate, so 3 phosphates are connected to it. That's how your body stores energy, or not stores, but uses energy in that form. How it releases that energy is the ATP-ACE will cleave off a phosphate off of the adenosine triphosphate, so it brings it from 3 phosphates to 2 phosphates. In that release, it's kind of like an explosion of energy being distributed in the area that it's at. So such as here, sodium, it's like this. Sodium comes and binds to it. High concentrations outside, or say here's the sodium channel. Like here's the membrane. Woo, okay, the sodium is on high concentration here, low concentration here. Here's the pump. Pump sitting here, and it's like this. ATP-ACE, the Na sodium potassium ATP-ACE pump is here. ATP is going to bind to it. It's going to cause an explosion as sodium goes into it. Sodium flows inside, hangs there, boom, explosion. It releases it outside, shooting into a high concentration. And the reverse for the potassium side. It goes from the outside into the cell. But why do we want it to do that? Because we have certain things that are called coupled channels, such as sugar. We want to get sugar into the cell. How are we going to get sugar into the cell? By a coupled channel. That sodium that we've been pumping, keeping a high concentration outside the cell, it's going to flow right back in through this coupled channel. So why are we doing it? It's a waste of time. Well, what it's going to do, that flow is going to cause sugar to flow into this coupled channel and help it pump itself into the cell, saving energy. It's a really, really good way that your cells do it. Your cells actually have millions of sodium-potassium pumps across one single cell. Then we have the last thing, which is the transport of large particles, endocytosis and exocytosis. Endocytosis is where we take particles that are so large they cannot go through a protein. And since they can't go through a protein, they're going to go through the actual plasma membrane. The plasma membrane itself is going to take it, bulge down, and then condense, and then just release it into the cell as a vacuole or vesicle. And that's going to contain it and transport it to wherever it needs to go. Reverse is exocytosis, where it takes it all, puts it in a vesicle, Golgi body puts it there, and it goes out of the cell and releases it out into the rest of the place. That's the end of this video. I shortened it, kept it as quick as possible. Thanks for watching. Be sure you watched this and did a good job. See you guys tomorrow or the next.